Okay. Good morning, everyone, and happy Friday. Okay, so let's start with some recap questions that will get us going from where we were last time and continue this time today. Okay. I think everybody was very interactive last time, which is good. Once we said there are participation points. So I will vouch for more participation points. So you're going to keep track, Sophia, with names? I will have them say their names. Okay, great. So what is the R group on an amino acid and how it may differ among different amino acids? Axel? Huh? Aerob? Oh, aromaticity. Yes, some would have aromatic structure. Correct. Yeah, Ali? Polar, nonpolar, basic, acidic. You gave 4. Great. Again, Axel. Sulfider groups. Very good. And there is also hydroxyl groups. So there is the extra amine group, basic, extra carboxyl group would be acidic, aromatic, aliphatic, hydroxyl groups, sulfider groups. The presence of all of these groups will impart reactivity of the different amino acids, will result in different functions. Okay, great. Name the 3 basic amino acids and the 2 acidic amino acids. Your name? Kate. Yeah. Yep. Very good. So remember those because these are very important because they carry charge and impact functionality quite a bit. So arginine, histidine, lysine are basic, glutamic and aspartic are acidic. I probably should put my mic on, right? Okay. I'll just do that. It's better. Okay, next question. How are biogenic amines produced? Give an example. All right, Lauren. Yes. Very good. So loss of a carboxyl group, decarboxylation. And example is tyrosine into triamine and histidine into histamine. Histamine will be present in spoiled fish, tyramine in fermented or aged cheese like the blue cheese. Good. Give examples of different reactions that involve particular functional groups in the R side chain. Tiffany? Yes, reaction with anhydride will give you a color compound for quantification. But specific R groups. Sarah? Disulfide linkages. Perfect. Alex? Yes, the carboxyamine group of glutamine and the amine group of lysine. Specify the functional group. What else? Can you give an example of post-translational change? Remember we talked about examples in milk, casein. Some of them are what and some of them are what? Due to what amino acids? Yeah, you can read into your notes. Yeah? Yeah, serine and threonine, they have a hydroxyl group. They result in post-translational changes such as glycosylation is one, and Justin? Phosphorylation. Okay, it's okay if you read from your notes. This is kind of a revision, so I don't mind. I don't expect that you know these by heart by now. So it's good that we're doing this. Let's see, did we miss anything? Good. What is pKa? Ryan? It's log of the dissociating constant. That is correct, but what is that point?
It's the equal amount of good charge and uncharged.
It's No, not the isoelectric point. We'll get to that. It's the pH at which we have equal amount of dissociated and non-dissociated. Let's say if we look at amine groups, if the pKa would be in the basic range. So at a specific basic range for a specific basic amino acid, you'll have equal amount concentration of NH2 to equal concentration of NH3+. So dissociated NH2, undissociated NH3+. Okay, so here's where we were last time. That was the last slide. We were talking about pKa. We're talking about the isoelectric point. The isoelectric point is what? Is the pH at which what? Zwitter ion. So the Zwitter ion, meaning you have no charge, no net charge. So you have equal amount of pluses and minuses. So that means they cancel each other, so the net charge is 0. That's the pH at which you have net charge of 0. The protein carries charge, obviously, but the pluses are equal to the minuses, so the net charge is 0. And then remember that acidic amino acids will have pK values at acidic conditions, and basic amino acids will have pK values at basic conditions. All right. So therefore, you have on any amino acid, you will have at least 2 pK values, one for the carboxyl group and one for the amine group. Okay. Sometimes you have 3. Why? So every amino acid will have 2 because you have amine group and carboxyl. Sometimes you have a third one. Why is that, Sophia? Yes. So the acidic and basic amino acid will have a third pKa. That would be due to their additional carboxyl or additional amine group. So here's an example of an amino acid, serine. So the pi of serine is equal 5.68. How was that calculated? How you calculate pi is you look at pKa 1 for the carboxyl group and the pKa 2 for the amine group. So let's say for the carboxyl group at 2.2 you have equal COOH amount to COO minus. At pKa 9.15 you have equal concentration of NH3 plus to NH2. So you have equilibrium here and equilibrium here. If you want to calculate the isoelectric point where you have net charge of 0, so you would add pKa1 plus pKa2 divided by 2, you get the pi. That's the standard way of calculating pi. But sometimes you have the charged R group. So you have the acidic amino acid and you have the basic amino acid. The way you calculate the pi for acidic, you take into account pKa1 and pKa3. That means the pKa for the carboxyl group on the R chain. For basic amino acid, the pKa3 is for the amine group that is on the R chain. The pKa for the amine group on the R chain. So here are the different pKa's. You need to memorize this by heart? No. It's like, what? You kidding me? No. But you just want to remember one thing or a couple of things. All carboxyl groups have pKa in the acidic range very low, 2, 1 point something. So because carboxyl groups are acidic, they don't want to give their hydrogen until there is a lot of acid in them, not give out, take hydrogen. Unless there's a lot of acid in the medium. You're titrating with a lot of acid and finally there is a lot of protons floating around, so they pick up hydrogen. So that's why the pKa is always very low. You really need to get to a low acidic pH for them to pick up the hydrogen. For the pKa2, the opposite. So amine groups, they don't want to give you the protons. They want to hold on to it until you titrate with a lot of base. There's a lot of OHs, OH minuses in the solution. Then they would hesitantly give you their protons. So you can see that pKa2 for amine group is basic. The pKa3 is dependent on whether the side chain is acidic or basic. So if it's acidic, like in the glutamic acid, it would be lower than 7. Aspartic acid is in the acidic range. And then the basic ones are in the basic range. Okay, and this is a list of isoelectric point. Again, it is also dependent on whether it's basic or acidic. You will see, for example, arginine is very basic, so it will have really high isoelectric point. Any questions about that? So that's what you really need to remember, pKa's, whether it's acidic or basic, at which range of pH it would be at. Okay. So we talked about acid-base. Let's talk about hydrophobic properties of amino acids. Hydrophobic properties of amino acids are very important for the formation of secondary structure. They are the driving force of the formation of secondary structure. So why is that? You know, we'll talk about free energy in a minute, but when the primary sequence of a protein is formed, and the primary structure is basically the amino acid in a sequence, that's the first structure, you have a lot of free energy, and we'll talk about that in a minute, but you also have hydrophobic residues and you have hydrophilic residues. That means you have hydrophobic amino acid and hydrophilic amino acid. Hydrophobic amino acids, by definition, they don't like water, right? So they just want to hide from water. So in their attempt to hide from water, they tend to associate with each other. So that there's a lot of free energy going on. So the free energy allows the protein to start folding, that sequence of amino acid to start folding. And what's driving the folding is the hydrophobic interactions between the hydrophobic residues, because they don't like water. So they want to hide from water. And in a cell environment, it's aqueous, right? So you have the protein is generated in a cellular environment. There's water around it. They don't like water. So in order to get away from water, they start to interact with each other via hydrophobic interactions, and the protein starts to fold. So you won't see a protein in its native structure as a primary structure. That is not possible. There's a lot of free energy, and there are hydrophobic residues that don't want water. So protein starts folding. So that's a primary role of the hydrophobic properties of some amino acid residues. Okay, we talked about amino acids and their properties and reactivity. So they are going to be formed into a polypeptide chain. So the bond that links amino acids to each other is known as the peptide bond. And it's a very characteristic bond. And it's a bond between the carboxyl carbon of one amino acid to the amine or the nitrogen of an amine group to the adjacent amino acid. So that's the peptide bond. And it's a condensation reaction because you lose one molecule of water when a peptide bond is formed. With each peptide bond formed, you lose one molecule of water. So it's a condensation reaction. So we call this N-terminus or amino terminus. And here we call either C-terminal or carboxyl terminal. You can hear you can see both terms. And this is the L configuration where you have the amine to the left. That's the natural configuration. So Defining the primary sequence or primary structure is the linear sequence of amino acids. That's the definition of a primary structure of a protein. So what dictates amino acid sequence is the genetic coding. So the DNA coding and then the messenger RNA and then ribozyme will produce the peptide or initiate peptide linkages. The average molecular weight of an amino acid is 135 Dalton. Of course, there's a range, but that's the average. But a protein is made up usually of so many large number of amino acids. So most proteins are in the range of 20,000 to 100,000 Dalton. So often we report them by kilodaltons. So we say 20 kilodalton to 100 kilodalton. So they're large molecules. So basically that's the message. Proteins are large molecules. So the linear sequence and length determines what? So the linear sequence of a protein and the length of a protein determines what? Or has an impact on what? In your opinion? Yes, Ryan? There you go. He summed it in 2 words. Yes, structure of the protein and the function. Structure of the protein in what way? So different amino acids, we talked about that. They are involved in different reactions and molecular interactions. So because they can form different molecular interactions, they will result in a unique structure. Based on the amino acids you have in a protein and the sequence in which they are in the protein, you end up with a particular unique structure. And with every unique structure, you have a unique function. And we'll talk about the impact of structure on function in later lectures. But yes, linear sequence, impact, and length, because also large proteins have different functions than small proteins. So these 2 impact structure, which in turn impacts function. So keep that in mind. How do we determine the primary sequence, composition, and sequence? So the primary structure is determined by, first of all, you want to determine composition of amino acid. How do we determine composition of amino acid? You break it down. You break the protein down to its amino acids by hydrolyzing the peptides. How do we hydrolyze or break down the peptides? By acid or base, concentrated acid or concentrated base. So the most common is 6 molar HCl at very high temperature, 110 degrees C, for an extended period of time to ensure that all peptide bonds are broken. Once all peptide bonds are broken, we run chromatography, which you'll learn about in food analysis. We run liquid chromatography, separate the different amino acids on a chromatographic column, and then we quantify each amino acid based on standards. So now we got the composition. How do we get the sequence? It's more complex. I will not go into that detail, but there are numerous ways of doing that. Edmond degradation is very common, where you degrade or you break bonds from the external exo reaction. That means you take 1 amino acid at a time. You break 1 amino acid at a time, react it with an Edmond reagent, and then the 1 amino acid goes into a chromatographic system and get analyzed. And then the next one, it's like a cycle. The next one gets broken off, gets analyzed. So you get the sequence that way by going from the outside to the inside. There's the nuclear magnetic resonance. You can get the sequence there. And of course, using mass spectrometry. Usually it's MALDI-TOF, which is matrix-assisted laser desorption ionization with time of flight. Again, you'll learn about these techniques in a different class. But there are ways to determine sequence of amino acids in a primary structure of a protein. So peptide bonds can be broken. We just said that you can break a peptide bond by hydrolysis with concentrated acid or a concentrated base. So we said 6 normal HCl is the common one. 110 degrees Celsius for an extended period of time can be up to 24 hours. You break that down into amino acids, but however, acid hydrolysis destroys different amino acids like tryptophan and cysteine. They are destroyed, and asparagine and glutamine are converted to aspartic acid and glutamic acid. So in order to determine these amino acids, you can do NOH hydrolysis. So oftentimes to determine the entire composition of the protein, you have to hydrolyze using different methods so that you can quantify everything. Peptide bonds are also cleaved by enzymatic hydrolysis. So there are proteases. So you'll have a lecture on enzymes, and proteases are one type of enzymes that are specific to breaking peptide bonds. But there are a lot of different proteases. And with that comes a lot of different selectivity. So you're probably going to learn about enzyme sensitivity and selectivity. Selectivity meaning, you know, the enzyme recognize a substrate by a binding site. So there's the binding site of the substrate, and there's the active site on the enzyme. So there's the active site and the binding site, like lock and key theory. So some enzymes are very specific. They just don't break any peptide bond. They want to see something in the binding site to latch onto it. For example, let's look at trypsin. Trypsin is a very specific enzyme, and it's present in our digestive system. It has to see lysine or arginine before it breaks the peptide bond. So if it sees lysine or arginine, then it will latch on to the binding site and break the peptide bond. Other enzymes like pepsin might prefer phenylalanine and leucine, but can also recognize others. So we call it general protease. So pepsin is another digestive enzyme in our gut. So it's more like general protease. You can hear sometimes if it's a general protease, not very specific, it can be called peptidase. Peptidase meaning it can't break any peptide really. So protease, if you hear peptidase, it could be a very general enzyme. So this is just for FYI, an example of different proteases and how they are differentiated in terms of preferred site and some sources. Okay. So moving on from primary structure to secondary structure to tertiary structure and then quaternary structure. So remember what I said about having a lot of free energy. So free energy leads to chaos. Chaos is conformational entropy. Probably seen delta G equal delta H minus T delta S before somewhere in your basic courses. So delta G stands for change in energy. Delta H is enthalpy. That's the energy contained in bonds. Okay? And T for temperature, and delta S is conformational entropy, which stands for chaos. A lot of free energy result in a higher delta S. Okay, so you have a lot of free energy and makes the molecule unstable. So the molecule by nature search for the most stable situation. So it wants to get rid of its free energy to become a stable form. In order to do that, bonds starts forming. Every bond gets formed contains energy. So the enthalpy increases. So containment of energy. So the bonds continue to form to reach the most stable structure. By that, you have mostly all the free energy contained as enthalpy. Now, remember this concept when we talk about denaturation of the protein. Denaturation of the protein is removing it from its natural form or changing its natural form. You do that by breaking bonds. Denaturation is a result of breaking bonds. So as soon as you start breaking bonds, delta S chaos becomes higher. Change in entropy becomes higher. Delta G becomes negative. So what I want you to remember, a negative delta G leads to instantaneous denaturation. And that is due to breaking molecular bonds and creating chaos. Okay, so we go from primary structure to secondary structure to tertiary to quaternary, and let's talk about how primary goes into secondary. This is what we talked about, role of hydrophobic residues. Let's talk about it a little bit more in detail. So, like I said, hydrophobic residues tend to interact with each other. They don't want water. So they start coming closer to each other via hydrophobic interaction, expelling water outside of the structure. So here's the water. It goes outside. So functional groups on the different proteins start forming hydrogen bonds. Instead of forming hydrogen bonds with water, now that water is expelled, they form hydrogen bonds with each other. So hydrogen bond is basically a hydrogen covalently linked to electronegative atom like oxygen, nitrogen, and sulfur. So, and then bonding with an electronegative atom. So that's a hydrogen bond. So like I said, instead of forming hydrogen bonding with water, they start forming hydrogen bonding with each other, and that would be, now two forces are taking place, hydrophobic interactions and hydrogen bonding. So the free energy is reducing, is going lower because we're forming bonding. Started with hydrophobic interactions and then hydrogen bonding. That is forming your secondary structure, is mostly stabilized by hydrogen bonding. So going from primary to secondary, you have a lot of hydrogen bonding going on. So if you are asked in an exam, what's the most stabilizing molecular interaction that would stabilize a secondary structure, you would say hydrogen bonding. If you're asked what's the role of hydrophobicity in the formation of secondary structure, you say hydrophobic residues don't like water, they interact with each other, initiating the folding process. Okay. So secondary structure, like I said, stabilized through hydrogen bonding. And you have two main forms of secondary structure. You have the alpha helix, which is usually formed, you don't need to know this by heart, but just FYI, the alpha helix is formed when you have this kind of sequence, polar, nonpolar, followed by two polars, followed by two nonpolars, so and so on. If you have this configuration, it's most likely you're going to form a secondary structure that is a helical, alpha helix. And then it's stabilized, you have the hydrogen bonds are forming like perpendicular to the helix structure. And then another characteristic feature is in every turn, you have about 3.6 amino acid residues in every turn. That's a good College Bowl question, if you guys are in College Bowl. That's a very common one. How many amino acids is in an alpha turn? It's 3.6. So beta sheet is another form of secondary structure. And it's formed if you have an alternating units of polar, nonpolar. And usually it is formed in such a way that within the same protein there would be strands. So the protein is going to form like kind of a strands within the same one polymer, and the hydrogen bonding, actually this is perpendicular to the axis, sorry. This is perpendicular to the axis, this is parallel to the axis. So the hydrogen bonds are formed perpendicular to the axis of the structure. Okay, so there are other forms. There's beta turn and there's random coil. Random coil meaning you don't have any particular shape of secondary structure. You don't have a certain sequence, and you might have a lot of proline. Remember, I don't know if you remember what I said about proline having this unique cyclic structure that makes it very rigid. We call that secondary structure breaker. So once you have a lot of protein in your structure, then you don't have a regular secondary structure. So secondary structure impacts certain characteristics. It impacts, for example, thermal stability. So let's look at these two examples of a protein. This is a whey albumin protein, and this is a beta-lactoglobulin protein in whey, or the whey is the liquid that gets separated from the curd in milk. So look at this. So arrows represent beta sheet, and these are the helical structure. So this one has one helical structure and a lot of beta sheet structure within it, secondary structure. The whey albumin, it's almost entirely alpha helix secondary structure. So a characteristics of that is beta sheet is more thermal stable than alpha helix. So this structure impacts the denaturation temperature. That means at which temperature the protein unfolds or changes from its native form. So you can see here beta-lactoglobulin denatures at 75 degrees Celsius, whereas albumin denatures at 64. So there's a difference. Denaturation impacts functionality. We'll learn about that when we cover denaturation, but that is an important characteristic. And another thing that is peculiar or unique, when you heat the solution of a protein that has alpha helix structure, when it cools down, it transforms to beta sheet. It becomes even more stable. So, you know, protein denatures, right? But then you have a lot of bonds that are broken, but while you do that, when you do that, you are creating free energy. So the protein would want to reform into another stable form, right? It won't stay open, unfolded. It will just reconfigure itself, forming new bonds. In this case, you go from alpha helix to beta sheet and becomes another stable form, but it's not the same as the original form. So that's why we call it denatured protein. It rearranges itself to another stable configuration. So again, secondary structure is not stable yet. You still have a lot of free energy. So more bonds continue to form. And then that alpha helix or beta sheet or beta turn or random coil will still continue to change in structure to get to the most stable form, and that would be tertiary structure. So tertiary structure is quite stable because you don't only have hydrophobic interaction and hydrogen bond, other interactions start to occur, like the ionic interactions, which is an interaction between a positively charged component on the protein or residue to a negatively charged residue. You form an ionic bond. That's another form. Another one, you can have van der Waals or dipole interaction. You can also have disulfide linkages. Disulfide linkages imparts the most stability because it contains the most energy. It's a covalent bond, so it contains the most energy. So if there are disulfide bonds, not all proteins have disulfide bonds, but if there are, then the protein is very thermally stable because it takes a lot of energy to break a disulfide bond. So usually proteins stabilized by disulfide bond denatures at high temperatures. Okay, let's look more closely about the molecular interactions within a polypeptide. So there are different groups. We learned about the different groups, and different groups will react differently. So let's look at first at the simplest form of interaction, or the interaction that contains the least energy. It's the dipole interaction, or it's also, you can sometimes see it as van der Waals interaction. So it happens between two neutral functional groups, and they have to be within a short proximity to each other in order to interact. And then forces are weak, so it doesn't contain a lot of energy, but it's additive. Because there are a lot of neutral components or neutral atoms, neutral functional groups, you will have a lot of van der Waals interactions. So although individual ones do not contribute to a lot of containment of energy, but there are a lot of them, so it's additive. So they do contribute to the stabilization of the protein structure. Hydrogen bond, we talked about that. It's the hydrogen bond between a hydrogen linked to an electronegative atom, and then bonding with an electronegative atom. So there's a lot of hydrogen bonding that occurs within the molecular structure. And these amino acids in particular, threonine, serine, aspartic acid, and glutamic acid, take part in hydrogen bonding more than others. So here, this statement states that the oxygen usually is a proton acceptor, and this hydrogen is readily gives itself. So there is the hydrogen donor, and then there's the hydrogen acceptor, which is partially negative, that attracts the hydrogen atom, and that's how hydrogen bonding occurs. All right, electrostatic interaction. This is another important bond that occur due to interaction, or we have repulsive forces and we have attractive forces. Repulsive is when you have two functional groups that have the same charge that are close to each other. That plays a role in denaturation. When I talk about pH later on and impact on denaturation, very high pH or very low pH will result in same charges. When you have all positive or negative, then you will break ionic bonding, and then the protein denatures. That's why low pH and high pH protein denature. So electrostatic interaction could be repulsive and attractive. In this case, it is attractive. You have an amine group in near proximity to a carboxyl group. Plus, minus attract each other, form ionic bond. It's also called salt linkage, salt bridge, or ion pair. All these terms mean the same thing. It's an attraction between a positive and a negative, forming an ionic bond. So these all charged groups can form ionic bonds. So of course, acidic amino acid and basic amino acid would be the ones that would form more likely the ionic bonds. Also cysteine and tyrosine, the cysteine has a sulfhydryl group and tyrosine has a hydroxyl group. At high pH, they become ionized. They give their proton. So they can also take part in ionic interaction, either attractive or repulsive. Hydrophobic interactions, we already talked about that. They don't like water, so they interact with each other via hydrophobic interactions. And this is between nonpolar groups, obviously. So because they are hydrophobic interaction, they are solvent dependent. Let's say when we talk about denaturation, we're going to talk about organic solvent. So organic solvents are nonpolar. So if you put the protein in an organic solvent, it's going to disrupt the hydrophobic interactions because now there's no more water. there is a nonpolar solvent in the system, then the amino acid will not necessarily interact with each other. They open up their interactions and interact with the organic solvent instead, and the proteins unfold, denatures. That's why, let's say we're extracting oil using hexane. If we keep that flour that has high protein in there and high fat in hexane for a long time, we're denaturing the protein. And that's why it's very careful in the soy industry where they're producing soy protein isolate. The exposure to hexane is very critical. All right, the last one is the disulfide linkage. Again, two sulfide groups in near proximity will oxidize and form disulfide linkage and contain the most energy, and that's the most stabilizing force in a tertiary structure. So again, not all protein has that, because not all protein has high amount of cysteine, so it might not form disulfide linkages. It's very protein specific. If they're there, then the denaturation temperature, as I said, would be higher. So that, because it's the most stabilized, I don't know what I did. It's the most stabilized, stabilizing force. Sophia, what's this? Okay. All right. So these bonds, this is a table that summarizes the bonds. It's a good table to study. So you can see here the covalent bonding, which is the disulfide linkages in this case, has the highest energy, contains the highest energy, followed by the electrostatic interaction or ionic bond. Then hydrogen bonding, followed by hydrophobic, and last is the van der Waals. They're not in order from highest to lowest, but you can see which is highest and the second highest. You don't need to know the distance here, but I would like you to know what functional groups are involved in each bonding. So cysteine, these are the amide, hydroxyl, phenol. Hydrophobic groups, the aliphatic and aromatic side chains, electrostatic, the charged groups, and any neutral groups would be involved. There are a lot of them. So there are a lot of van der Waals. Again, you want to know the disrupting solvent. So covalent bond, you can break it by reducing agents. Because it's a bond formed due to oxidation, you can reduce it and break the disulfide. So you can use reducing agents, and there are a bunch of them: mercaptoethanol, dithiotol, DTT for easy pronunciation, sulfide, cysteine. They're all, if you add them to your protein solution, they're going to break your disulfide linkage. And it's common that we do that for changing the functionality of the protein or for analytical purposes where proteins are large. You just want to break them down so you can look at subunits individually. So there are certain places where you actually need to disrupt these bonds. Hydrogen bonding are disrupted by urea because urea will form hydrogen bonding itself with the components of the protein, thus breaking the hydrogen bonding within the residues. Same with detergent, they would have the same effect. And heating. Heating disrupts hydrogen bonding. When we talk about collagen, you know, collagen is a protein that will be formed into gelatin, and it's used for gelation. So collagen, when you heat it, it will break hydrogen bond and then restructure itself to form gelatin. The different reconfiguration is gelatin. So this is an example where hydrogen bond is broken by heat. That's the thermally induced gelation of meat protein. Hydrophobic bonds are disrupted also by detergent and organic solvent. The detergent that most commonly we use to disrupt these bonds is STS, sodium diacetyl sulfide. You don't really need to know that, but some of you are familiar with this detergent. Electrostatic, so changing the charge disrupt ionic interactions. So if you increase the pH or decrease the pH, you're changing the charges on these residues. If you add salt, salt will interact with the charges on your residues. So the Na positive will interact with carboxyl groups. The Cl minus will interact with the positive group. What did I say? Na plus interact with negative. Cl minus interact with positive. The other thing I wanted to mention is heating disrupt hydrophobic interaction. Heating enhances hydrophobic interaction while disrupting hydrogen bonding. Cooling enhances hydrogen bonding. So you want to know that the impact of heating is the opposite on hydrogen bonding and hydrophobic interactions. Okay. So here are different structures. So you have the beta-casein and you have the beta-lactoglobulin. Beta-casein is one of the caseins in milk, and beta-lactoglobulin is one of the proteins in whey. So the beta-lactoglobulin has a globular structure. It's a compact globular structure because it's composed of a relatively equivalent distribution of polar and nonpolar. So the nonpolar tend to reside in the interior and the polar tend to reside on the exterior. And the distribution is homogenized. There are not pockets of hydrophobic and pockets of hydrophilic. It's homogeneously distributed in the protein, so it results in a compact structure. Whereas beta-casein is amphiphilic, meaning it has kind of a tail and a head. So you have a location where it's very hydrophilic and a location of very hydrophobic, that's what makes it a good emulsifier. In addition to that, it has high percentage of proline. Having high percentage of proline makes it more of an open structure. You have random coil as a secondary structure, and then you have more of an open amphiphilic structure. So this makes it, sodium caseinates are used in industry mostly as emulsifiers because of their amphiphilic nature, whereas whey protein are mostly used in beverages for their great solubility, because on the surface of this beta-lactoglobulin you have a lot of hydrophilic amino acid that will interact with water. So here's an example of structure impact function, where the tertiary structure states that beta-casein is emulsifier, whey protein is excellent, has excellent solubility and used mostly in high protein beverages. Okay, in many cases, it doesn't end at the tertiary structure. Stabilization is needed in a quaternary structure. Quaternary structure is when you have individual polypeptides interacting with each other to form a stable quaternary structure. We talked about the casein micelle last time. We talked about how the phosphorylated groups are on the exterior stabilizing the colloidal system in milk. Why this is needed? Because caseins have a lot of hydrophobic residues on the surface. If they are just present like that, they're going to interact with each other, not wanting to interact with water. So they need to be stabilized in milk. So nature, Mother Nature, created something to make them stable in milk rather than get the milk and the casein precipitate. So Mother Nature created this colloidal system where you have the quaternary structure of the casein stabilized by phosphate linkages and the hydrophilic phosphorylated group on the outside stabilizing it. So it was necessary for these proteins to form this quaternary structure to be stable in milk. So sometimes we need that. So we need the quaternary structure. Whey protein doesn't need that. Whey protein does not exist in a quaternary structure because it's hydrophilic on the surface, interacts with water, doesn't need to be stabilized further. This is another globular structure, which is the hemoglobin. And it is basically 4 peptides or polypeptides, and you have the iron within that hemoglobin. So it's a protein that carries iron in our blood. So even quaternary structure can be different. You have the fibrous proteins like the collagen, so and also actin and myosin have a fibro kind of shape. Whereas the globular, you have these individual proteins that are globular in shape, and they interact with each other and remain globular in shape. So I think this is the last, I will just take 30 seconds, finish that. So when you have a fibrous structure, that means you have a lot of polar residues, and they interact via hydrogen bond. And if you have cysteine in there, they can be stabilized by disulfide linkages. So if you have this structure, the fibrous structure, that means you have a lot of polar. They want to interact with water. They don't want to hide. If you have a lot of residues of the hydrophobic, they want to hide, and they form this globular structure. And so the fibrous structure is mostly structural protein, collagen, actin, myosin, or the globular are usually enzymes, and they're also functional proteins. Okay, I think I will talk about this slide next time. Thank you. Oh, one last thing. I'm recording these lectures, and Sophia is going to put them on the Moodle. So if you want to go back, you'll find them there.